Hi Shanice, I'm sorry I missed your call. I was working on the paper last whole night. I didn't sleep because the editorial company is in Switzerland. So I wanted to get it over with. So we have different timeline. So it's a little bit tiring day. So I missed your call. So tell me, okay, how are you? How is your thing, mom? What kind of stroke? What was in the brain? Oh, I'm so sorry to hear that. And then that they operated. Did they do the surgery? Mhm. Soft food. So she's conscious. She's okay. Okay. Oh, thank God. Thank God. Mm. And then the physiotherapy and everything will start, right? Physiotherapy and everything will start. So did you see her on the screen or something? Hmm. So she was still positive. Right, right. And you're positive too. You accept. Okay. So hopefully they will vaccinate her also when she's all right. Yeah, there is sometimes tough time knocks without any, you know, not any awareness, anything out of the blue. But good thing is that good care. So how far you are from Jamaica Medical School Center? 15 minutes walk or drive? Drive, okay. So a little bit distant. Mhm. Okay. Well, I'm glad that things are things will improve. Yeah, I understand that. It's yeah. It's tough. But so far you're in good hands. So, yeah, thank you. Mm. Oh, you're welcome. How is your prep going? So when you say these exams, you will report your score. How much they expect from you? Mhm. Mm. There was one lady in my, I think for 2019 molecular biology class or 20, I don't remember her name. I think somebody was telling that she got there, Emanuela. Mhm. Yeah. Okay, keep working hard. Yeah, it's the last few, and sometimes these things happen when you need most focus. But I think these times test you and make you stronger, more focused. So yeah, so I would say take this message in your uh, strength and just gather your energy and do the last lap. That's what it matters. So good luck and feel free to call anytime. Okay. Take care. Take care. Have a good evening. Okay. Bye. Take care. Bye.
Hello, Professor.
Yes.
How are you doing? I want to apologize first for last week. If I seemed a little short when talking to you about my quiz, I just was very stressed out and concerned. So I apologize for that.
Okay.
I just sent you an email regarding quiz 1, and the screenshot in the email shows that both tests were submitted 5 minutes apart.
Yeah.
And the only reason I made it a second attempt was because it seemed to offer me a second attempt, and the instructions did not say that the time would be attributed to the time I had left in the first attempt. So as due to the submission times, you can tell I didn't get through the test. So I was just hoping that you can give me the score of 21 from my first attempt, which is the completed exam, as opposed to the 4. And if you have any issues, I stated in my email, as far as being able to change something, I can contact Mr. Wymore if that creates less work for you. And I could ask him to figure it out, or whatever I need to do.
Yeah, we should go into why it happened, because contact Windmore and say why it happened, because I only give instructions, and there are certain of checklist that I have to complete on the blackboard. So that's at my end. Now the same blackboard is at your end that I have no control. My intent was that each student get to attempt one question twice, right? Now, how this works out at the end of different students face different sort of issues, and I have no control. So please contact Wymore and understand what is going on, because
my confusion is I don't understand what you mean when you say you have no control, because I know it says in Blackboard.
Because as I'm again saying, and I don't have time to go over it again and again, that there are certain things that I checklist. For example, I say that the number of, let's say if I say there are 20 questions, and then I say one question gets 1 minute. So I put 20 minutes. Right, but sometimes some students get late submission. So I don't know why that system is behaving like that.
Oh, I didn't have a late submission.
I'm just giving you various examples. You have to let me speak. I am listening to you. And because unless I put my thing across, you will not understand, and I cannot go back and forth over the same thing since last week. You sent me an email. I will look into it. I mentioned to you last week, and it is okay with me to have you get away with 21. Okay.
So you're fine with giving me the other grade.
Yes.
You just don't know how to fix it in the system.
No, I don't know. I don't have any.
Okay. So when I contact Mr. Wymore, I could tell him that
I don't want to be in that loop, because that's between you and the Blackboard administration.
Okay.
I'm repeating with you that, let's say if I give 10 questions and I give 1 minute per question to each student, and I say that each question a student can take 2 attempts. Now, let's say how does the time varies, and I give time from 7 a.m. to 11:59 a.m. I don't know those things. I just give the command checklist, take this time, attempts, questions, random, one screenshot, and then that's it. How does that appear? Why does sometimes different students have different experience? I have no idea.
So when I say this to you, you don't know why it stopped.
No.
You don't know why it just submitted and didn't let me finish my second attempt.
No, I don't know.
Okay, but I should have been able to.
I don't know. As simple as that.
So when I talk to Mr. Wymore, so that I'm clear, I can tell him that you're fine with giving me the 21, and he just needs to figure out how to switch it over.
Correct? All right. Okay.
So that's fine to tell him. I don't want to misrepresent you to him.
I have nothing to worry about.
No, not for you to worry about. I'm just not
I
clear.
think
I'm not clear.
I just mentioned to you that I am okay with 21 and that's the end of the thing, right?
Yeah, yeah. I just want
And
to make
whatever
sure.
the Blackboard is behaving, you could talk to the Blackboard administrator, and that's another kind of discussion.
Okay. And I sent you the email, so you have documentation of that.
Okay,
So
when I get a
that's
chance, I will look at it.
okay. Thank you very much.
Welcome. क्यों नहीं खजा रही हो? Hi, how are you?
Hello, Professor.
How are you?
I'm okay.
How is it going?
It's going on okay. But for the last assignment that we sent in today, well, we're supposed to send in by 11:59 tonight. I uploaded it, but it was the wrong assignment I uploaded.
Okay, I didn't get a chance to look at it. It's fine.
Okay, is there any way that you could like erase it and I could upload the new one?
No, I don't know how to do that.
Okay, so how would I do it? Like send it to your email?
What happened exactly? Tell me again. I couldn't like I couldn't get the complete story.
Okay, so I uploaded the assignment that is.
So hello class. This session today is almost we are in the middle of March, and every week we are making a little bit of progress and trying to cover the syllabus. In today's topic or today's lecture is focused on stem cells, and stem cells is one of the most upcoming cutting edge research in the field of biology. And why I say that, let me give you an example. So one of the major problem that human society is facing is organ failure. Right? So let's assume that somebody is suffering with kidney dysfunction or chronic kidney disease, and there are some organs in the body, unfortunately, which is very difficult to treat if the disease is chronic, like liver cirrhosis or some imbalances with kidney or heart even. Almost all organ, if something happens and something goes wrong that cannot be controlled, it starts to deteriorate. And so let's just take focus on kidney failure. So let's say somebody has a kidney failure. So what are the options? I think the only options after taking the medication and coming to the conclusion that yes, kidney is going to fail, so a person would need transplant, right? So what is a transplant? Transplant means you get a foreign kidney, right? Foreign kidney or a foreign organ. Now, even if you're getting it from your sibling, it is still a foreign. But advantage is that at least in the sibling, some genes are matching as compared to somebody who is totally unknown, who would be a donor. Uh, in a transplant, what happens? A donor donates the failed organ to the sick person, healthy organ to the sick person. So it is better that the tissue, it is called as tissue typing. I don't want to go into the detail, but that after tissue typing, there should be a significant match between the donor and the recipient. And it is expected that sibling, two kids from same parents will share those genetics. But despite that, they may not be 100% match. So what happens here? The kidney initially get rejected, and that's the stage where there a person becomes susceptible to many infections and so that's a so one problem is organ failure. The second problem is to find the right donor. Third is to deal with acute rejection because of the foreignness of the organ. So what is happening now? Things that are happening now is population is increasing, right? And a lot of people, because of many reasons, there is high rate of organ failure, right, as compared to and kidney failure. So there's a long list of patients who need kidney, and it is very hard to find a suitable match. So in this situation, what can happen that physicians can or physician scientists, those people who are MD and also PhD, and they're working on stem cell and organ regeneration or tissue regeneration. They can take patient cells, and they can train these cells to become kidney, right? And these kidney can be put into. So what is the point I'm making here? So the person whose kidney failed, cells can be taken out from the same person, and it can be developed into a healthy kidney, and this patient can get his kidney back. Now, as much as I said it like sound very easy, there are a lot of challenges. It is still a budding field, stem cell biotechnology, but that's the hope, to get stem cells to become a factory to produce or to replace diseased organs. Because in coming time, when the population is going to be more, people are going to be more sick, there has to be an alternative path. And any alternative path is more challenging with regard to the is that place that you recognize how miraculous a human body is in terms of putting together various specialized organs and make our body function like a factory, and every organ is important. So that's to make the long story short, stem cell are those cells that can be trained in the lab to regenerate or generate an organ that can replace the sick organ, right? So I gave you one scenario, but there can be multiple scenarios. Let's say there is somebody got an accident and hurt his brain or important organs. So, and it's hard to find a replacement. So this will be, if this becomes the day of, if this field sees the day of the light of the day, and it becomes, it offers to people who are suffering, it will be a great thing. So that's what technology is about, to always beat the boundaries, always push the boundaries, and that is for two reasons: human health and to take care of the diseases. So healthcare is going to change significantly. It has already changed. You see, one virus, it has changed the whole world, and so many problems that we come to know that a virus can generate. So think about these chronic illness. They may not be pandemic, but they are very serious. So today's talk or today's lecture is about stem cells. So let me take out the slides. So stem cells can be used for many things. So for example, brain stroke, there can be a local local injury to the brain, and that can deprive the brain of oxygen. The oxygen level is very critical. Brain is one of the most metabolically active organ in our body, right? And it totally it's dependent upon proper supply of oxygen. And let's say if somebody has a heart attack and the heart is stressed, the pumping of blood is stressed up, then what is happening? The blood is not going into the brain and the blood is not taking the oxygen. So on one hand, that person will have heart attack, but could also have brain stroke because oxygen is not delivering. On the same line is traumatic brain injury, right? Trauma could be, a mild stroke, and accidents, right? So these traumatic brain injury, traumatic means it cannot be strong or it cannot be weak, but it is strong enough to create sort of signaling in the brain that changes, that has a ripple effect in the brain that causes hypoxia, oxidative stress, and so on and so forth. So as I mentioned to you, brain is most metabolically active. It is very soft tissue, and there are some age-related diseases. Also note that the pattern of diseases are changing, right? For example, diabetes. So diabetes is a disease of hormones, insulin, right? But you know, so diabetes is because of insulin. The insulin is produced by pancreas, and within pancreas there are two kinds of cells, alpha and beta. Beta cells are responsible um, for production of insulin that controls sugar in our body. So if there is a degeneration of beta cells, then how to get beta cells? That is a very specialized cells. Pancreas, very specialized, sophisticated, delicate organ. So the only way is to generate islet cells. And then you don't want to get islet cells from somebody because it is going to cause rejection and it can cause pancreatitis. So it's better to have islet cells from the patient, him or herself. So again, muscular dystrophy. So as simple as hair transplant, right? So a lot of people who have less hair, they go for hair transplant. So I don't know the details of hair transplant, but what is happening that they're implanting the hair follicle such that the hair comes out again. What are those hair follicles? Hair follicle has all the characters of a stem cells. Why do I say that? So why do I say that? Because our hair grows few inches, per month or per week. I don't know. It depends from person to person. So how does it happen? So we take monthly or bi-weekly or weekly haircut, and again hair comes same as nails, right? We keep clipping them, but they keep coming back. What is that force that keeps our nail to keep growing, hair to keep growing? So those hair follicles, right, are type of stem cells, right? So actually hair transplant is a sort of a stem cell transplant. And then again, blood cancer. All the blood, it emerges from bone marrow, right? And bone marrow, you will see such a complex architecture of bone marrow. There is one mother cell, that's bone marrow stem cells, that gives rise to so many kinds of cell that we will see. And any kind of cell, any one type of cell can go bad, and it can cause cancer or anemia, right? So there are anemia. Anemia means lack of iron in the blood, and that could relate to lack of less RBC, red blood cells. That could relate to less hemoglobin. There could be plenty of reasons, but it all comes down to bone marrow. So there are some diseases like anaplastic anemia, in which bone marrow is not healthy enough to produce these kind of cells, and that has huge effect on the growth of an individual. right? Bone marrow, somebody's anemic, then if somebody's anemic, then more susceptible to infection. Then wound healing, right? So let's say skin, so melanoma, it happens in the skin. And if it happens in the skin, so it's visible. So people recognize melanoma because we, as compared to some organ inside our body. So melanoma if detected at right time, it can be taken out. And once melanoma is taken out, it can cause a big loss of flesh from the skin or from the upper part of the body. So wound healing or plastic surgery or skin transplant, all those things, the inspiration to those things are stem cells. Okay, so what are the basic ways to isolate, to work? So the work on stem cells are being done actively in labs, right? And these labs are very sophisticated. To work on stem cells, it's very expensive. Uh, no matter we think that, okay, cell is a fundamental unit of life, so what? It's a single cell, but it is a box of mystery. It's so difficult to control a cell in the lab. So why do I say that? For example, I take out a skin cell. So a skin cell in our body will be destined to to become skin cells. For example, let's say if we get a razor cut, after a few days, you'll see that wound from the small razor, small cut has healed up. Why? Because the skin cells became skin cells. Same philosophy, same thing is very difficult to reproduce in the lab condition to make skin cells to become skin cells. How does our body work? We still don't know. What controls the fate of the cells in our body? We still don't know. And we know a lot, but we need to know more. So young people like they need to get into become a part of research workforce. Young people come with new ideas. So coming back to the class topic, so stem cells will be isolated from the same person. Why? To not have that effect of rejection. So take the cells from the person who is sick, take the healthy cells from the person who is sick, put it in the petri dish in the lab, provide the nutrition media. So medium means it contains nutrition. Now, believe it or not, every cell, every tissue in our body, they need different kind of nutrients. A brain cell will need different kind of nutrient as compared to heart cells. So, let me just tell you a short story that I was working um, on cardiac ischemia, right? And we used to isolate myocardial cells, cardiomyocytes, from the heart of the mouse. And we used to grow it in the Petri plates. So, these cells that we used to grow in the Petri plate, that used to beat like heart. They used to, you can see through the light microscope that those cells are throbbing inside the Petri dish. And then again, you take a cells which are cancer cells. They will grow so aggressively. They will not beat like heart cells. So the point I'm saying that our cells are very unique. And because of that uniqueness, it's very difficult to treat all the diseases with one medicine. So you need to go into specifics. And organ failure is one of the rising illness. So you get the cells from the patient, grow it in the lab in a petri dish, try to provide the same environment that is inside our body. That's the only way you are going to make skin cells or kidney cells into kidney cells. You have to know, we still don't know much, what is the environment inside our body that is directing the path of these cells to become specialized. And then once they become specialized, then we try to do some research on the stem cells to make sure that if we want skin cells, then it is a skin cell, and then put it back into the patient. So this is a very broad path to elucidate the use of stem cells in clinic. And it is very much going on, especially with the help with so I can say that bone marrow transplant is one of the most regularly used stem cell techniques to take care of diseases. Now, I think in my almost all lecture, I have made a point to touch COVID. So how can stem cells help COVID? So you know that some people develop very severe lung infection, so much so that their lungs have holes, and a lot of other diseases. And nobody knows at this point of time what is the inflammation, why so much of a degree of problem with lungs, and why different people have different symptoms, and why people are getting holes in the lungs. So how to punch those holes, how to take care of this problem, right? So another problem is to stem cells, because again, you don't want to take cells from other person to avoid rejection. So scientists can take out blood from the body of the patient, take it to the lab, try to purify those cells which are, this is most critical. So blood have millions of cells. How to purify those cells, how to isolate those cells which will transform into lung cells, and these lung cells can be brought back to the patient so that that hole in the lungs can be closed, and a person can go to daily life. So these are the potential treatment of potential clinical and research use of stem cells. So now let's learn a little bit basics about stem cells. So there is a hierarchy in our body, right, that we should appreciate. Our body is not made in a very disorganized way. It is very organized. So let me just explain to you how it is organized. So we all, start our life, most living beings, including human, we start our life from zygote, right? Zygote is formed by the fusion of male and female gametes inside the mother body. So what is this zygote? This is a single cell, right? This is a single cell, right? So much easy to work with, but we still don't know. So what is the programming inside this zygote? This zygote has the story of our whole life, right? And all those things are present in the DNA and the proteins in the zygote. We still don't know much about zygote, because this is the zygote that has the capacity, one single cell that has the capacity to develop into a complete human being or any organism, that zygote. Now this zygote, which has a tendency to develop into any cell, any organism, this kind of potential is called as totipotency, meaning that the zygote has the tendency, the power, the potency comes from the word potential. So zygote has total potential to develop into an organism. Right? But when after the zygote is formed, after fertilization, the cells has to divide very, very in a timely bound, time sensitive way, because inside our mother's body, all human beings that develop into a complete individual in 9 months, the clock is very, very fixed. And if there is so if there is any delay in this clock, or there is any dysregulation in this clock, then there is a potential cause of some disease to mother or to child. So this is the most miraculous thing that happens in human being, the motherhood, right? How a mother can carry a child, and a child that comes from the fertilization of male and female gametes. Just now I told you that if something foreign goes into our body, it can reject, but motherhood is totally different. right? So try to appreciate the human body, and in itself, it's a miracle. So it's the development of human body or human being is timed 9 months. It has to be totally done. Delay or advancement, not good time. It's a concern for physician. So after the zygote is formed, cells starts to divide, right? So what happened that these cells, they form 3 layers after zygote. These 3 layers are called as ectoderm, mesoderm, and third endoderm. So as the name suggests, that these 3 layers, the mesoderm is the middle layer, ectoderm is outside layer, and endoderm is inside layer. Now, these 3 different layers, right, they're not only layers, but they have capacity to develop into, to have the footprint, to have a path to develop into different organ. Now, this capacity of 3 layers within a human body or any multicellular organism, these 3 layers to develop into 3 different cell fate, which will give rise to whole organ, because our body is a multi-organ body. So these 3 layers can give rise to all the organs that is needed for our body to survive. This is called as pluripotency. Pluripotency. This capacity is called as pluripotency. Okay. So what would ectoderm develop into? Ectoderm will develop into outside its outer layer of cells, skin cells. It can develop into nerve cells. We'll look into later. The middle layer can develop into bone marrow to give rise to blood cells. It can develop into muscle cells, and so on and so forth. The endoderm is responsible for development of all the internal organs. For example, digestive systems, right? Circulatory system, respiratory system. All these are developed by endoderm. So, but the capacity of ectoderm, mesoderm, and endoderm to give rise to all these spectrum of organs within our body, that power is called as pluripotency. Okay. Now let's just consider one of these cells from here. So let's just consider bone marrow cells. These bone marrow cells can give rise to many kinds of cells, but let's look at 3 different kinds of cells. This bone marrow cells can give rise to 3 kinds of cells. One is RBC, which is called as red blood cells. WBC, it is white blood cells, right? So when we go to a clinic or for our physical checkup, they always look at RBC. And RBC, the number of RBC give rise to our hemoglobin level. And WBC, they will look at various other cells to look at our capacity to fight infections. And the third type of cells is platelets. Now, one mother cells, bone marrow, giving rise to 3 different kinds of cells. And these are different because RBC carries oxygen, WBC fight infections, And platelets, they help in clotting of blood, right? What if we get if there are diseases, right, where people have tendency that their blood do not clot because they have low platelet count, thrombocytopenia, low platelet count, so they have clotting problem. But the point I'm making here is that um, bone marrow cells, which emerges from the mesoderm emerge from zygote, give rise to three types of cells. One more type, but I'm not going to the details. RBC, WBC. So three different kinds of cells. This tendency is called as multipotency. I hope you are noticing that what did I mention before I started going into this? That there's a hierarchy in our body, right? So from totipotent to pluripotent to multipotent, from zygote to three layers to bone marrow, and then all these cells. And all these cells, the development in our mother's body happens timely, right? If it doesn't happen, if RBC is not developed inside the fetus at the right time, then who will carry oxygen in the fetus body? And then if there is lack of oxygen in the fetus body, then it's an urgent situation. So RBC has to develop inside the fetus body, inside mother's body, right at the right time, right? Because mother's body is providing oxygen through placenta. And if RBC is not developed inside the fetus, it's a problem, challenging situation. So this is multipotency. Now think about WBC. I mentioned to you about WBC, that these are the cells that helps to fight infection, right? They help to fight infection. So what are these cells? These cells are lymphocytes. 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 Then you have basophils. Then eosinophils. Basophil, then you have neutrophils. Neutrophils, right? So these are different kinds of WBCs that try to fight infection, right? Lymphocytes can fight viruses. Basophils, they fight parasitic in tropical countries. Kids, they have worm infection because of not good water, not good quality water and food. So basophil tries to protect them. Eosinophils is directly related to allergy. Neutrophils, they help in fighting infection. For example, let's say if we get a splinter in our finger and then it develops into a pus, that pus is what? That pus, it comes from when the neutrophils kill the bacteria. So this ability of WBC, to make these kind of cells, WBC comes from bone marrow. This is called as multipotency, right? Multipotency. Now let's take one example here. Let's just talk of lymphocytes, right? I just mentioned to you that lymphocytes help in fighting infection. It could be bacterial, it could be viral. Now these lymphocytes, they are of two types, T cells and B cells. Right? These two cells, so T cells, they try to produce a protein called as cytokines. And B cells, they produce antibodies. These days, you must be hearing the word antibodies a lot in terms of post-COVID infection, whether somebody has antibodies or not. So who produces that antibody in our body? That's B cells. And where does B cell comes from? It is a type of lymphocyte. And what is lymphocyte? It's part of WBC. And WBC, where it comes from? Bone marrow. Where does bone marrow comes from? It comes from mesoderm. Where does mesoderm come from? It comes from zygote. So again, I'm trying to make a point that there is a very, elegant hierarchy in our body. And in the end, the last example is I want to give is, and that's much more related to this chapter called as unipotent cells. Unipotent cells are those cells which are part of very, very specialized organs. Right? Specialized organs. Like, for example, brain. The brain cells divide very slowly, and the division, the rate of division um, slows down with age. Same with liver. So if something goes wrong, then it always very difficult to treat. So some cells can only grow into that. For example, liver cells. These liver cells are called as hepatocytes. Right? These liver cells, they grow very slowly. They're extremely mature. They take a lot of time to grow. So that's why if there is a liver damage or liver cirrhosis, transplant becomes a very obvious choice. And then again, I mentioned to you, transplant is a challenging thing to find a donor. So stem cells is best to develop as a therapeutic. So this is the message was that our body is a hierarchy. It starts from totipotency, pluripotency, multipotency, and then unipotency. So this one thing perhaps I missed, that the tendency of lymphocytes to develop into T cells and B cells, this is called as oligopotency. So let me repeat, totipotency, pluripotency, multipotency, oligopotency, and unipotency or unipotent cells. So this is the way our body is organized and we function. So let's go back to the slide. And this is what is shown in this slide and that I described in the last on the whiteboard. What type of, now let's come to stem cells. So what are stem cells? So stem cells are those cells, right? These are very unique cells, stem cells. They have two properties. The properties are that they can divide and they can self-replicate. They can divide and they can self-replicate. So self-replicate, and self-replication means division. And then they have a capacity to differentiate into many cells. And this capacity to differentiate into many cells comes from the specific environment. If you give these stem cell environment to grow RBC, they'll grow into RBC, and so forth. So let's look at the difference between what is stem cell differentiation versus renewal. So by definition, stem cell differentiation is a process by which normal cells develop into specialized cells. So our body is full of specialized cells. There is no extra cell in our body. It has got functions to do, right? Extra cells can be a problem and it can give rise to disease. Self-renewal means it can divide indefinitely. It can divide indefinitely, right? It is possible that the rate of division might decline with aging, but again, it has the tendency to divide. So what's the effect of differentiation? It can give rise to functional and structural properties. For example, in case of lungs, if the scientist is making lung cells, so treat a COVID patient who has lung problem, they have to train the cells to develop into lungs. And what is the function of lungs? Exchange of gases, carbon dioxide and oxygen, right? So these are our cells have structural and functional properties, right? And what is renewal? So the ability to regenerate. So that's the difference between differentiation and self-renewal. Now, where is stem cells? Where are stem cells formed? So stem cells are formed in a fifth day embryo. This fifth day embryo is called as blastocyst. Fifth day embryo is called as blastocyst. A blastocyst, you see here, it's a spherical shaped structure. Spherical shape. This is fifth day. Within the blastocyst, there are two things. A space called as blastocele, and there is a small population of cells. They are called as inner cell mass. This inner cell mass is actually your stem cells. This is your stem cell, inner cell mass, right? And this, so think about it. If a scientist is working on stem cells, he has to take out these cells. So he has to tamper with the embryo. That's why sometimes working with human embryonic cells is controversial. Because yes, unless these cells are taken out from human embryo, scientists cannot work. Human embryonic stem cells derived from human embryo, right? So to just review this slide, stem cells are found, human embryonic stem cells. So there are different kinds of stem cells. One of the most common stem cell is human embryonic stem cells, which is found in the human embryo. And this is found on the fifth day of the development, called as blastocyst. Within the blastocyst, there are some specialized cells called as inner cell mass. They are very, very naive cells. They haven't seen the environment. They haven't seen the population, pollution, or different environmental hazards as compared to adult cells. So these are very naive. They can be developed into any cells that a scientists want. But the challenge is that we don't know everything that is happening in our body or our body requires to differentiate into specialized cells. So let's look at this slide. And as I mentioned to you, so just to give an example, this is human embryonic stem cells. This human embryonic stem cells can develop into ectoderm, mesoderm, endoderm, and they can develop into various progenitor cells, right? Neural stem cells will develop into neural crest, glial cell progenitor, neuron progenitor. Mesenchymal cells can give rise to muscles and osteocytes, the bone cells. And hemangioblast from mesoderm give rise to hematopoietic cells, which has some type like bone marrow cells. So there is a hierarchy in our body, right? And this is exactly our body develops inside our mother's body, right? If any stage there is a delay or a failure, it's a potential problem to the growing embryo. So this slide also is showing the same thing, multipotential hematopoietic stem cells. This can give rise to two kinds of progenitor, myeloid and lymphoid progenitor. Right? Lymphoid progenitor can give rise to lymphocytes into T cells and B cells, and B cells can mature to produce antibodies, and that mature B cells is called as plasma cells. Then this is a natural killer cell. The other myeloid cells, it give rise to RBC, erythrocytes. Then myeloblasts, right? It give rise to basophil, neutrophils, eosinophil, monocytes. And then thrombocytes, which are platelet cells. So there is a hierarchy in our body, and especially look at the fascination of blood cells. The life cycle of blood, RBC or lymphocytes, depends from individual to individual. It could be any time between 3 to 6 months. And then after that, our blood cells are replaced by new cells. So this production of blood in our body, when I say blood, I'm taking everything into account, the whole picture into account. So our blood cells keep recycling, the turnover every 3 to 6 months. Now, remember one thing, when I say 3 to 6 months, it's not that, let's say today's 16th March, so all the cells will be, turned new into, let's say June or July. No, Different cells, they grow at different pace. They are at various different stages. And that is advantageous to the body because they have something there, right? So it's not like 100% or zero. So now here I want to tell you that I mentioned to you different kinds of cells, right? How do you know that this is, okay, RBC, they are red, so you can see, like, when we donate blood or when a phlebotomist take out our blood, we know it looks red, so it's RBC. But how do you look into lymphocyte T cells, B cells, growing muscle cells, how they differ from each other? How do scientists work? Because unless they have the power to, they have the technique, they have the tools, technology to let's say take out heart cells to take care of heart attack, or to take brain cells, neuron cells to take care of brain stroke. We have to know. So each cell has its ID, right? So we all have our ID, like social security number, right? Or a driver license, which is our picture ID. Likewise, cells also have an ID on themselves. This is called as cell receptor. This ID on the surface of the cell is called as cell receptor. This cell receptor is present on cell surface. So an RBC will have a different receptor, a lymphocyte will have different receptor. Between lymphocytes, T cells and B cells, they have different receptor. Those are their IDs, cell receptors, right? That's how scientists can isolate, let's say, B cells from the pool of blood. So my PhD was on HIV/AIDS patients. So I used to work on the blood of these individuals, and I used to look at the lymphocytes, because the lymphocytes presence or absence or low number could correlate to the disease. If somebody is sick or AIDS or having many infections, so how do I used to work on lymphocytes, because these lymphocytes have a special ID. So B cells was their receptor had CD19, CD19. The T cells, they had CD4 and CD8. So that's the point I was trying to make here. Now, this can go as complicated in this slide, where you see that how within the blastocyst and human embryonic stem cells, right? Human embryonic stem cells can develop into primordial germ cells, and then it can develop into gonocytes, and then premature germ cells. And when the puberty comes, it can develop into female or male gametes. So, right. So what do you see here? So this is a lineage, right? Going from left to right. And at each lineage, there are markers, which are receptors. For example, OCT4. So if you use OCT4, you can get cells like the human embryonic stem cells, PGC, gonocyte. After this receptor is absent. So then if you want to work on gonia cells, then you have to work with stellar receptors. If you want to work on mature germ cells, then you have to work with nanos. So these are IDs, right, present on the cells. And we know the kinetics of these IDs on these cells. As the cells are developing, they are changing shape and function, size. So look at this, blastocyst. And then how does human embryonic stem cell inner cell mass looks? Then how does it look when it develops into primordial germ cells? Then gonocyte, then gonia. So cells keep changing their shape and size. And how do they change? They're all imprinted in their DNA, right? We still don't know what are those mystical features in the DNA that helps the cell to go through this lineage. So to work on stem cells, we have to be master of these things. So let's look at this slide. So let's focus here. Look at this. So this is bone marrow. This is bone, and within the bone there is bone marrow. And if you look at the bone marrow has an architecture. It's just not fill of something like filled with something like soft blood or soft flesh. It has an architecture, adipocyte, bone matrix, so on and so forth. So the cells that are supposed to form blood, It will emerge from bone marrow and become hematopoietic stem cells. Then it can give rise to lymphoid family, it can give rise to myeloid family, and then the bone cells itself. Okay. So remember, now there are many cancers, right, where cancer, if not treated, or if it is very difficult to treat, cancer, they spread over the body. That's called as metastasis. And how does the cells flow from each other? The blood cells, blood flows to every part of the body. So it's part of blood or lymphatic system that the cancer cells metastasize. So metastasize. So sometimes what happens, that bone marrow or the bones get affected, right? Why? Because the blood cells and the bone precursor cells, they all emerge from the bone marrow. They all emerge from the mesoderm, right? Now, as I mentioned to you that, what did I mention to you? I mentioned to you that these cells, they have passport, they have IDs, right? And those IDs can be used to isolate these cells. Why do you want to isolate these cells? To develop into treatments. So for example, human hematopoietic stem cells or hematopoietic stem cells, these have CD50, 150 positive, CD48 negative, CD. So what is CD? CD stands for cluster differentiation. Cluster differentiation. These are IDs. These are cellular receptors present on human hematopoietic stem cells. So if I want to work on these cells, I have to isolate these cells. So make sure I use these IDs, right? Passports, visa, to work on these cells. Similarly, multipotential progenitor cells. So this is CD150 positive, this is negative. This is 48 negative, this is also negative, but this is 244 positive. So this presence of 244 positivity makes it a different kind of a cell. Then again, leukemic stem cells, right? Where does cancer emerge from? They emerge from the same bunch of cells, right? Something went wrong. So in this leukemic stem cells, what happens? It is still CD150 negative, but it becomes 48 positive and 244 positive. So if they have these two kinds of IDs, means that it has a potential to become leukemia, leukemic cells, blood cancer. So unless we know the knowledge of these things, we can't develop treatment, right? So this is one of the treatment for many blood disorders. So let's say patients with spinal injury. Sorry for this, diffuse slide. So if this person has a spinal injury, then it's very difficult to put somebody else spinal tissue. So take the bone marrow from this patient and train this bone marrow to become mesenchymal cells, cultivate them in the petri dish in the lab, and then put it back in the patient. Now, I mentioned to you that stem cells has controversy because to work on human embryonic stem cells, one has to puncture the embryo to take out the inner cell mass. So, so many scientific development, it also depends upon government policy, public opinion, debate, and all those things. So when scientists felt the burden that, okay, working on human stem cells is unethical, then what to do now? So scientists started working on adult stem cells. Now remember, human embryonic stem cells, it has derived from the 5th day embryo. So those cells are very soft, very naive. They can be easily educated. But adult stem cells, it's very, very rigid because it has been trained to an environment. It's difficult to change course. Naive cells, you can train. Adult cells, they are already educated. It's hard to turn them around, but still scientists are trying. So if neural stem cells, they are found in brain, and they can give rise to glial cells, neurons, bone marrow cells, platelets, RBC, WBC, mesenchymal cells, and I described. So even in both human embryonic stem cells as well as in adult, bone marrow cells are bone marrow related diseases. It could be cancer, leukemia, anemia. They are more successful clinically as compared to other diseases. So look at these key terminologies. What is embryonic stem cells? What are pluripotent cells? And then what are the differences between stem cells, adult and human embryonic cells? I repeat, I'm sorry, human embryonic cells and adult stem cells. So one is naive, one is from the adult tissue. One is human embryonic cells is from embryo, fifth day from blastocyst. Adult cells are from specific organs. So if somebody wants to work on neural stem cells, they have to get the stem cells from nervous system. It could be brain, it could be part of neuron, so on and so forth. Now, a little bit information about how to grow these cells in the lab, right? So the idea is to provide them the same environment that they are having in our body, right? And in our body, there are, if you have studied BIO472, there are so many kinds of support system. For example, blood, it brings a lot of nutrients, oxygen. Uh, the neighboring cells through autocrine, through paracrine ways, they give hormones, growth factors, so on and so forth. That's how the body of an embryo develops inside the mother successfully, because they provide the nutrient, right place, right time, for right moment, for right duration. And that's very difficult to recapitulate inside the lab on a Petri dish. So to provide nutrition is providing a good environment, right? Good, so one of the ways scientists they use to grow stem cells is so what they will do, let me go back to the whiteboard. What scientists do to grow stem cells is very difficult. So what scientists will do, they will take a Petri dish. Let me take a different color. So scientists will take Petri dish. I'm trying to draw to help you understand. So let's say this is a Petri dish. So what scientists will do, they will take first the mouse fibroblast, and they will seed the mouse fibroblast on the Petri plates. The whole plate will be full of mouse. And these cells, they are called as feeder cells. These mouse cells, they are called as feeder cells. Right? So what they will do, they will secrete various kinds of growth factors. And on this, scientists, what they do, they overlay the stem cells, human embryonic stem cell or adult stem cells. They would overlay. So these are stem cells. So who is feeding them? It is being fed by mouse cells. You can ask me a question that why not human cells? Why not so this is a tactics that if you use cells from two different species, you can distinguish between them. If you use human feeder cells and human stem cells, it can become confusing. So if you get a question, the feeder cells in the stem cell regeneration in a lab, it should be from same species or different species? It should be from different species. For example, mouse here. excuse me. So these stem cells will grow in this petri plate, and then it can be brought back to the clinic for treatment. So this is a timeline for the development of the field of stem cell technology. So it started as early as 1975, right? To look at mainly at hematopoietic stem cells. Then first embryonic stem cells isolated from mouse embryo, right? And then this mouse embryo was first conversion of adult rodent fibroblast to myoblast. Myoblasts are those cells that can form muscles or then in 1995, the sheep was cloned, Dolly, if I remember the name correctly. And then, right? And then first human embryonic stem cells were harvested in 1995, right? So every 5 years, there's a major discovery, major, milestone achieved in the field of stem cells. First clinical trial in 2007, 2008 to spinal injury patients, right? And then first clinical trial, I will come to this in the end, iPS cells, iPSC cells, induced pluripotent stem cells. So things don't happen in one day, it takes time. Again, in iPS cells, from mouse, they moved to human clinical trial from 2006 to 2016. So now I am going into iPS cells. What are iPS cells? These iPS cells are called as induced pluripotent cells. So iPS cells, IPSC stands for induced pluripotent cells. Now I have already explained to you the meaning of pluripotent, right? A cell that can give rise to multiple lineage. What do you mean by induced? Induced means you give a trigger. You give a trigger to the cells, right? And when you give a trigger, the cells react. And one of the ways cells react is to develop into next phase. So how this triggering happens. So let's say now, first of all, why this technique came into being, why these cells came into. So what was scientists they were facing? They're facing difficulty to work with human embryonic stem cells. They're facing difficulty to work with adult cells. So they were constantly looking for new things. So remember, when we lot of cells are constantly shed in our body, healthy cells. For example, our digestive tract, they keep shedding, outer layer once in a while. So those cells that we pass through urine also, those are healthy cells, but they come off our body and they pass through. So those cells, now scientists are thinking that they can be trained to develop into stem cells. So when I use the word training, means induce them. So you take the cells, for example, let's say if we take the cells from, let's say, skin cells, right? We keep shedding skin all the time. Or so let's say, let's take the skin cells, and then This skin cell, how to trigger it? We trigger it through addition of 4 different genes. 4 different genes. So when you put 4 different genes, these genes, they trigger, what do they do? What they do? They trigger different kinds of proteins, right? And this protein, they transform into skin cell or skin fibroblast, or let's say urinary cells that we pass through our urine, right? Or what we pass through gastrointestinal tract. These cells can be induced with these 4 genes, and they can be trained to develop into stem cells. Now, what are those 4 genes? Those 4 genes are these ones. These 4 genes, they are called as reprogramming factor because they can reprogram the cells. Right? So very important. OCT4, SOX2, KLF4, c-MYC. These are 4 different genes. When we put these genes into the cells, they can acquire the property of pluripotent stem cells. Right? So the scientists who did this, who found these 4 genes that can transform a skin cell, mouse embryonic fibroblast, to become stem cells, these scientists, they got Nobel Prize. So that's the trigger. These 4 genes, they trigger to make factors that would transform the regular cells into stem cells. So induction. So this slide also shows the same thing. KLF4, SOX, c-MYC, NANOG. If these genes are put inside the cells, they acquire the property of stem cells, they become reprogrammed, and they can be trained to develop into various kinds of cells. And you know, this picture is from a company called as R&D Systems. So they work with these kind of ideas. Right? So same concept I'm giving here. These are somatic cells, they're cultured and they're transformed with this 4 genes, and they can be used for developing therapy, right? They can become sick disease cells, and these disease cells can be treated with different kinds of molecules. For example, let's get a fibroblast from skin cells and develop into somatic cells, stem cells. And these stem cells, they mimic leukemia or skin cancer or melanoma. So you can use this cancer cells, which is induced pluripotent cells actually, to find out new medicine, right? So same. So these 4 factors, they are transcription factors, OCT3, 4, KLF4, SOX2. So think about from where we started this chapter. We started talking about COVID, treatment, organ transplant, then we went into the hierarchy of our body, and see how science has jumped up from 1975 to 2020 or 16 for healthcare. So in eyes, the stem cells is very successful. Macular degeneration or you know, where cells can be taken and it can be cultured in the lab and it can be put back to the eye. Then also one of the biggest challenge of is Alzheimer's disease, because we don't know when does this disease start, because it is such a slow disease. We only come to know when the damage is already done. So compared to healthy brain, the Alzheimeric brain is small, shrinked. So only way we can do this is by treatment, and it can be done by stem cells. Now, one of the theory that is coming up, or has already come up, that some of the cells, some of the stem cells, when they become abnormal, they acquire cancer kind of a phenotype, characteristic, and they keep growing, right? So the hypothesis is that normal cells, normal stem cells, because of certain mutation or radiation from normal stem cells, they become cancer stem cells. Because people think, why do cancer cells keep dividing, right? Just like hair follicle, right? So there also must be having some stem cells. So those stem cells which support the growth of like irregular growth, aggressive growth, uncontrolled growth in an individual that give rise to tumor or cancer or metastasis. There is no differentiation. They become just like dividing, no control, right? So these are cancer stem cells. So this is the difference between the normal stem cells and cancer stem cells. So they can initiate tumor, metastasis, and they're very heterogeneous. Cancers are not, sometimes they can be homogeneous, but mostly heterogeneous. That's why it's very difficult to kill the cancer cells because they're not alike. So I believe with this, we finish our chapter on stem cells. So this will be recorded and